Okay, so every 2 minutes, somewhere in the world, a woman is diagnosed with breast cancer. And just within the brief moment that it's taken me to come here and start my presentation, yet another cases have been added into the list. So within this staggering, I'm sorry, can we start a slide again? I think it's some. I want to start it again, sorry. This is the last slide.
She wants to start from the.
Okay.
Is it the one?
Yep.
Okay.
Thank you very much. Sorry for the delay. So if we look at this staggering statistic, we can see that there's one subtype of breast cancer called HER2 positive breast cancer, which account for 25 to 30% of all cases. And it stands out because of its aggressiveness. And if we have a closer look at this subtype of breast cancer, there are defining features of this subtype is the overexpression of HER2 receptors, and this can drive the rapid progression of the disease. And in order to address it, there's efforts have been made with the development of HER2 targeted therapies and drugs like trastuzumab or pertuzumab that target the extracellular domains of HER2 receptors, or some other drugs like lapatinib or neratinib that target the intracellular kinase activities, have transformed and opened a new era in breast cancer treatment. However, this progress has been tempered by a critical issues, and that is therapeutic resistance. So this is when microRNA could come into play, but the question is how. So microRNA, it is a small non-coding sequence, and after biological processes, it can bind into mRNA sequence, and it can either inhibit the mRNA translation, or it can either degrade them. And in both cases, it is considered the regulators of gene expression at post-transcriptional level, and due to that, it can influence cancer progression and, of course, the therapeutic response. So we could see the potential of microRNA in here, yes, obviously, but then we look at the current status of the study into microRNA that regulate drug resistance in breast cancer, and we have found that the majority of them is focusing on microarray analysis technology. While this technology is invaluable in microRNA profiling, but the limitation of it causes the result to be still incomplete and fragmented within individual studies. And there's another a broader possible issues that remain here. If we have a look at this figure, we could see that there's only a minimal overlap between mRNA findings across the study. And due to these, we have another issues that we want to overcome. How do we have a final general conclusion of all the whole picture of microRNA that regulate drug resistance in HER2 positive breast cancer? Because as we can see from now, our understanding of the mechanism is still incomplete and fragmented. So our study, we deal with this by investigate, of course, the mechanism of HER2 drug resistance through microRNA analysis, and we do based on data obtained from the public database, and we do bioinformatics analysis, and we do meta approach to overcome the issues of limited overlap microRNA findings between different studies. And we then do functional mapping with the microRNA when we do functional analysis for downstream evaluation. Okay, so pretty straightforward, the method that we've been using here. For first part, the data manipulation. We do first, we have a systematic search between the along with all the mRNA, microRNA datasets in the GEO database, and with all the inclusion criteria that we set up that regulate drug resistance in HER2 positive breast cancer, we managed to have 3 microRNA datasets, and for these we do data acquisition for using an R package, and we do pre-processing, normalization, and we do differential expression analysis, and we do the next step is meta-analysis, and we use combined effect size to get the list of our potential microRNAs. And finally, we would do target prediction, because as I mentioned before, the microRNA would bind into mRNA and that regulate the therapeutic response. So we would predict the target gene that microRNA binds. And for external validation, we do analysis in mRNA datasets, and with the same process, we do process and normalize data, and we do differential expression analysis. And finally, we came up with the list of the differential expression gene. And for the further step in functional analysis, we would overlap these 2 lists, and we do some further gene ontology analysis, KAGG pathway analysis, PPI, and we also do clinical relevance evaluation using survival analysis, and we use the protein data from the human proteome atlas. Excuse me. So for differential analysis in microRNA datasets, we managed to finally have 113 differentially expressed microRNA. And for mRNA external valid datasets, we have managed to identify differentially expressed gene from each of these 3 mRNA datasets. And for the overlapping, we come together with 110 genes that was identified to be both differentially expressed and are predicted as target of those differentially expressed microRNA. So in functional analysis, if we have a look at this first figure here, we do gene ontology analysis for both biological process, cellular component, and molecular functions. And we managed to come up with some critical terms that involved, and we identify those genes that belong to those terms. And we would next do KAGG pathway analysis, and this figure shows the 10 most enriched pathways in our analysis. And as we can see here, there's some pathways that is very common in breast cancer, and like estrogen signaling pathway, and we also identify key genes in those pathways. And I wouldn't bother to show another complex PPI network in here, but on together we do put the PPI network result on with these results, and we come up with a final hub genes that is considered to be potential candidates for further analysis. And we do prognostic values and clinical evaluation of the hub genes, and we have, as we can show here, this is the result from the survival analysis. And for these 2 of the hub genes that are significant in the set, and we also have a look in their human proteome atlas profile, and we can see here's the result from the cancer tissues, and we have another one from the normal tissues, and we could see that there's a high intensities that have been expressed for this gene FOS in cancer tissues. So yeah, on together, we would have, there's some limitation in this study. First of all, we include, even though we have a systematic approach in this one, we've in screening all the data sets and stuff, but in this field, there's still limited number of sample size within those data sets. And also, we are still lacking of experimental validation in this field, but we did manage to identify biomarker and targets that may enhance the effectiveness of this targeted therapy, and into finally get a suitable treatment strategies for patients with HER2-positive breast cancer. So therefore, in the future, we would love to have a larger and well-curated data set for further analysis, and we would also continue with our experimental validation of the findings. And yeah, my name is Hoa, and I would like to thank my supervisor, Sweta, Adele, Ola, and Jay for their advice in this project. And I also would like to acknowledge 2 clinical oncologists, who is my clinical advisor, Dr. Emet and Dr. Isam. And this study is funded by SUTU President Funding. And that's all. Thank you very much.
Thank you for the excellent talk. We have some time for questions, so please feel free to approach the mic stand.
Hi, thanks for your speech. And I have a question is about the microRNA prediction, because I know there are several programs like the Target Scan and the Mir system. So I wonder, how did you choose it? Because I used to use the same program, and I find actually the results from them are not accord.
Thank you. So just to be clear, the question is which tool that I use to predict the target of microRNA is it?
Sure.
Okay, it's a multi-mirror because I think it's quite common in the field at first, because I came across a lot of studies that have been using the tools for the prediction of microRNAs, and also it's not only just the predicted result, but it's also a combination of all experimental validated data, and it's a combination of results from 14 database and datasets related with experimental validated data for the mRNA. So
I'm wondering, do you have validated these results by other program like the Mir Bridge or something else?
Yeah, we did think of like an overlapping between either that tool and some other tools. But when I do initial overlapping, I do feel like this one have yield a highest result, and I don't want to miss on any potential microRNA. That's why I decided to go with this one.
Okay, thank you.
Thank you very much.
Hello, thank you for the interesting talk. I was just wondering whether you observed any correlation between the mRNA expression as well as the expression of its target gene. So for example, if the expression of a particular mRNA is going up, the expression of its target gene should concomitantly go down. So I was wondering if you observed any, and if you did, what was the strength of that correlation? Thank you.
Okay, yeah, thank you very much. It was very interesting. Yeah, we normally we know of Fem microRNA as the silence of that gene. But I guess what you mean to ask is finally, what would I get if I identify that microRNA as a biomarker? And it can either enhance the resistance or either it will reduce the resistance. So based on the fact that it can regulate that gene, and we can further do some analysis of the gene, we could see, okay, if it inhibit that gene, whether the resistance gone or whether it become more resistant to the drugs. And that's the purpose of the thing, of the work that we do.
Thank you.
Thank you.
Yeah, great work. Thank you. My question is about Go analysis and the pathway analysis that you mentioned. And Richard does this enrichment analysis that only looks at the number of differentially expressed genes that fall on the pathway, but ignores the position and the role of the genes, the direction and the type of the signals, and so on. And gene ontology sometimes gives you too many biological processes, let's say, that are significant. So the first question is, how many biological processes came up as significant after the correction for multiple comparison in your case? So that would be the first question.
Okay.
Ballpark, not the exact number, right?
Yeah, after the ones I couldn't recall exactly how many of
it's
them.
Like 10, 50, 100?
It's yeah, it wouldn't go to 100. Yeah.
Oh, somewhere between 50 and 100?
Yeah,
Okay,
around, yeah.
thanks. And the second question is, did you try to look at some of the more recent, more sophisticated ways to do the pathway analysis, like impact analysis, which looks at the way the perturbation propagates on the pathway and so on?
Yeah, that would be a nice suggestion. I will make sure that I have to come back and have a look into some more extensive tools.
Great work.
Thank you very much.
Yeah, sometimes I'm just going to hop in with one question of mine. So sort of following up on the question by one of the audience earlier, when you did the overlapping, was the direction of the differentially expressed considered, or just a pure overlapping by the gene name, so to speak?
Yeah, it was just overall overlapping because I don't want to get, like some assumption before I get the final result. Just so, yeah.